Good evening, everyone. I'm Dr. Mamatha, Assistant Professor, Department of Life Sciences, Genetic Biology. Today we're discussing about C-binding technique of chromosomes. So before understanding C-binding, we should learn the basics of karyotype. Karyotype is the complete set of chromosomes of an organism, where the chromosomes are arranged on the basis of their shape, size, structure, and line. So we humans have a total of 46 chromosomes, 22 pair of chromosomes, given the name, the number now, 22. And in females, there is X chromosomes. In males, we have X and Y chromosomes. Also, in organism, many banding techniques have been used. So, C banding is one of them. C banding, which stains the constitutive heterochromatin, which is present mainly around the centromeres of albumin chromosomes. And it is most abundant around the centromeres of chromosome 1 and 916, and also at the distal part of the distal arm of the Y chromosome. The main principle of C banding includes denaturation and reassociation of the base pairs of the chromosomes. So during the C banding procedure, chromosomes are treated with a denaturing agent which causes DNA to unwind. The highly repetitive DNA sequences present in heterochromatin will reassociate faster, and the results in darker staining pattern when we stain with the gypsum stain. The pre-treatment includes treatment with acid. The acids like HCl can be used for the treatment of metaphase chromosome, so which removes the purines in the chromosome. Then it is followed by alkaline treatment. The barium hydroxide can be used for the alkaline treatment, which mainly reduces the ap-related sugars. Then finally, the hot salt treatment or hot salt wash are given using saline sodium citrate or SSC. Okay, so here the highly repetitive sequence which are resistant to breakage and renature. Okay, this is the major principle followed in C binding technique. So, as I mentioned, the chromosomes are obtained from metaphase plate, and they're treated with alkaline to denature, and sodium saline citrate treatment is given at 60 degrees centigrade. So then it is stained with Giemsa solution. So once the Giemsa solution treatment is given, we can see banding patterns. So these banding patterns we can see at heterochromatin region of the chromosome. And this is the C banding of human female chromosomes, where we can see a darkly stained centromeres of every chromosome. So this helps us to identify the genes which are located near the centromeres. The main applications of C banding includes identification of chromosomal rearrangements. So any chromosomal abnormalities like translocation or inversion can be identified easily using C binding techniques. And it also helps us to study heterochromatin variations, like the links between heterochromatin variation and the phenotypic triads. Okay, these are the important application we can study on the using C binding techniques. Then karyotyping analysis, like to understand the chromosomal numbers and structure of the chromosome and chromosomal abnormalities can be understood by using C banding techniques. In the species identification and evolutionary studies can be done by comparing the chromosomal centromeres of the chromosomes of different species or organisms. And investigating sex chromosome differentiation. The Y chromosome can be studied using C banding techniques. and study plant chromosomes. So using G banding or Giemsa banding technique is difficult to understand plant chromosomes, but C banding techniques will help us to understand uh, the structure and arrangement of chromosomes based on their size, and will help us to understand plant chromosomes as well. Thank you.